Hi, everyone. I'm Jamie Panetta. I'm the program director for the San Diego Independent Scholars Organization. So I want to welcome you to this talk. This is actually our last talk for this before we take a break for the summer, and then we'll start back again in September. So if you are not a member, I don't see too many non-members here. If you're not, if you're interested in SDIS and you want to get more information, please sign in and we'll send you some information. Okay, before I introduce our speaker today, just a couple of things. Let's hold all of your questions until the end, unless you have some very pertinent clarification question that you want to ask. This talk is being recorded, and so everyone online, sorry about that, everyone online will be muted. When we get to the questions, you'll have to unmute yourself to ask the question. And then the talk will be posted after the lecture on our website, which is SD Scholars, so San Diego Scholars.org. You'll get access to the video recording, okay? All right. So we are very fortunate today to have our speaker, Dr. Margarita Behrens. Her talk titled Epigenomic Control of Brain Maturation and Aging. A few words about Dr. Behrens. She obtained her PhD in biochemistry and molecular biology from the University Autónoma in Spain. She is currently, as I understand it, research professor at Salk Institute and part of the team that's creating the single cell epigenomic maps of the mouse and human brains that is funded by the NIH Brain Initiative. She has a background in genomics, neurobiology, and physiology, and she studies the balance between activating and inhibiting neurons in brain circuitry, primarily in the prefrontal cortex, which, as you know, is an area that's our executive system responsible for decision-making and reasoning. And today she will discuss her work in relation to brain maturation and aging. Dr. Behrens.
Well, thanks a lot for the invitation. Please let me know if what I'm talking goes over your head and ask questions, even though he said wait for the questions. If you're getting lost, just raise your hand and I slow down. If I don't talk everything that is on the slides, it's fine. But the idea is to try to pass the concepts of what we are doing. So it's not moving.
It did before. How do you tell you to change slides?
It's not moving the slides.
No.
Oh boy. Let's see, can I do it? No.
Yeah. Let's try.
Here we go.
Yeah, just taking a little bit time. Okay. Oh, what's happening? Oh, goodness.
We obviously need a scholar to upgrade the electronic.
We need to upgrade something.
We checked it out and it was working before.
We saw you do it.
Okay. And let's
Full
see.
screen.
Maybe if you put full screen.
Yes, let's play from the start here. Okay, and then let me make sure that the Zoom is still. Is it on Zoom? Is Zoom working?
Yeah, no,
Okay, so
you have to do share screen and.
Yeah.
Right, but. Oh, the other thing is, excuse me, sorry about that. We're losing power here. And where's our power forward?
There's one behind the table there.
Ah, right here. And we had all this worked out.
Apologies, everyone. We tried this all out ahead of time and now there's a last minute split. It was working.
Oh, no, this is.
Maybe,
not
maybe
good.
while we're waiting,
No, it worked.
can the speaker, enlighten us as to what epigenomic is?
Yeah, I was
Or will
thinking
you
of
be-
doing that because I just got a question. So
Ah.
epigenomics and epigenetics are
It's
same
not
word.
reach.
Epigenetics is usually for defining phenotypes, changes
Hand it,
related
hand
to
me,
changes in traits that are not dependent on change in the DNA, but they are more related to environment, exposures, and that do not change the sequence of the DNA. When we talk about epigenomics, we are referring to analysis of the DNA itself and how those epigenetic marks happen in the DNA without change. Basis. And what we try to make the difference, we are looking at behavior, we are not looking at the phenotypic changes of the organs or subjects, but we are looking at the modification of the DNA itself. And other, which I'm gonna talk about, you have in your body, every single cell in your body contains the same DNA, isn't it? The sequence, usually when you do a 23andMe, you work your with PBS and send it out,
Control.
and they sequence your gene. All your cells in the body contain the exact same genome.
Mhm.
it's locked in
However,
my car.
the cells producing your hair
No, the
do
power.
not get confused and start producing fingers. So you have a expression of the DNA that is particular to every single cell type.
I'm sorry, the power to turn that projector on, where was that?
Where did he put it? He probably put it somewhere here.
Oh, my goodness. No, I'm sorry, let me go find.
I thought he had put it
Is
there.
that the other computer?
No, it's a remote control.
Anybody's pocket, maybe?
It's kind of big.
Probably went back to the.
Anyhow,
Can you hear me? Zoom people. Yeah,
Yes, I can hear you.
okay, so you
well,
will-
sorry, we are having technical issues.
something happened with the computer,
It
not the
disappeared.
Zoom computer.
He put it back.
Oh, okay. There.
So basically, the way your genome, which is exactly the same in every single cell of your body, expresses differently in every cell type you have. And that way your cells do not get confused. When do they get confused? In cancer,
Turn it on.
which is when they regress from that state of differentiation and form what is similar to a stem cell that starts reproducing and aberrant. But that is the only time where you have that change. Otherwise, your cells do not forget who they are.
Okay.
And how they do that, how do they do that control is regulated by epigenomic effects, which I'm gonna try to explain to
If we can get this going.
Well, I can just,
I think we're back, and let's see,
yes. Success,
maybe.
yes,
It's,
yes, it is.
okay.
So, where we are gonna try to talk about this big brain here, our brain. But to get to it, we are gonna go through several steps that will help us get into this brain. So just for oh gosh. So as you all know, different functions of the brain are concentrated in specific regions of it. So in the frontal cortex, this part here is where executive functions are controlled. And as we get to the temporal lobe is where basically where your hippocampus is and memory is formed. And so both regions contain a very similar cellular composition, but the functions are very different. And so the question that we have is, are these differences due to intrinsic cellular identity so that the cells that are in the frontal cortex are different from the ones in the temporal lobe that control memory? Or are these functional arise from specific connectivity patterns? This is an image of a where they trace the connectivity patterns around the brain. And as you see, these are the processes and where the cells connect in different parts of the brain. And so it's a balance not only on the cellular composition, but who is talking to whom in the brain. And so do these specific connectivity patterns change the cellular manifestations, or are they stable? And once that connection is made, that is the way it goes, and the cells remain similar. And so to understand the brain cellular composition, we go back to animal models such as the mouse or the rat, where we can go and start defining. And so when we look at cortex, for example, we have several different, I don't know how much you know about the organization of the cortex, but the cortex, which is the big part of the brain that has expanded enormously from for example, the mouse, it grows in a very characteristic pattern. So the first cells that appear in the cortex are the deep layers, which are the layer 6 neurons here, and they have a very specific function. And then it grows outwards this way. So in the fetal brain, the first precursor cells appear here, and they give rise to these cells first. Once that layer is finished, the cells, which are called radial glia, start making the next set of cells. So it's an inside-out manner. And the last ones to be produced are in the layers 1 and 2. Layer 1 has very few neurons, it's more connections, but the layer 2 has very clear. That is for what is called excitatory neurons in cortex, the one that transmit through glutamate and are like the accelerator in a car. Then we have of neurons that control these accelerators, which would be the brain. And those are the inhibitory neurons. Those ones are born in a different part of the brain, and they migrate and come in to that cortex that is developing and position themselves in very specific part. And functionally, there are about 16 types of these neurons, but in general, you can group them into neurons that come from a different part of the niche that produces them. Those same inhibitory neurons that are produced, that region, which is called the medial ganglionic or the ganglionic eminences in development, gave rise also to all the GABAergic systems in the brain. So striatum, nucleus accumbens, etc. All their original niche is in the ganglionic eminences. And so when this is the set of inhibitory neurons that you can find in if you cut across a slice of the cortical tissue. And these are the different types of excitatory neurons that appear in the different layers. Each layer has a connectivity pattern that is proper from the layer. So on the deep layer neurons usually are projection neurons, and they are the ones that make help bring the thalamic inputs into the cortex. The top layers usually receive inputs coming from outside the cortex and coming in. So it's a whole circuitry that is formed early during development and then matures throughout life. And it takes, as I said, about a long time to reach maturity. In fact, it is believed that it takes to get a mature brain between 25 and 30 years in the human. Of course, we cannot study this in humans, but we are trying to. So the questions are, how many types of neurons? Believe it or not, we have a knowledge of the classes of neurons, but how many in each class and how they differentiate themselves, we didn't know. What are the similarities and differences across different regions of the brain? So you find, for example, in the cortex, you have a layer 6 pyramidal neuron in the frontal part of the brain. How different it is from a layer 6 pyramidal neuron in the visual cortex. They both belong to a cortex, but we know physiologically speaking that they behave different, but how much is that difference? We didn't know. How stable these differences are? Can we identify alterations in their development? Can we use epigenomics to study this? And so that's what we were talking. What on earth is the epigenome? And so to give you a little background, when the human genome was sequenced, everybody thought, oh, we are done. This is what we know. We are still mapping it, but we are gonna identify disease causes, genes. We are gonna have personalized medicine. We are gonna even advance the sequence technologies to find out more, and it is the foundation for understanding the blueprint of human beings. Good. But that's not all. As I was explaining, all your cells have the same genome, but they are not expressing it at the same time. And so here you have, for example, in plants, you have a mutation in DNA methylation machinery, and you can see here, these two plants look very different. However, their genome hasn't changed. In bees, the genome of the queen bee is exactly the same as the genome of a worker bee, but they are not the same. The reason why they are not the same is because the queen bee is fed a special food that contains high level of methylation substrate. And it's a methylation change in the queen and not in the workers that make this different. And in mouse, you have the agouti mutation that gives the different color to the fur. And again, the genome is not changed, but the color and shape of the animal is changed. And that is what is called epigenomics. I will go a little further, but from the same genome, you can have all these different cell types that cohabit in your body. Well, these are in many different organisms, but you give rise to neurons, to skin cells, to limbs, to all in the same. And so it's that manifestation of the genome that we try to study. And so the phenotypic complexity is a manifestation of transcriptional programs encoded in the genome. So those transcriptional programs are not the same for every cell type. They are particular to each cell type. The way the genome regulates this expression is through processes that regulate transcriptional programs such as transcription factors, non-coding RNAs, regulation of enhancers, and all those epigenomic modifications that allow the system to manifest itself in this particular way. And those epigenetic process, for example, covalent modifications of the DNA. So the basis, the sequence of bases of the DNA is not changed, but you can covalently modify the DNA and allow it to shut down or express itself. And then modifications in the histones are also going to allow you to cut the DNA or expose it for being transcribed. And so how can we study the epigenome? In the last few decades, we have found many ways of, and this is, I have been participant on some of the technology more recently, but this is old stuff. So here you have RNA being produced in a part of open chromatin. So you have all the different components of that chromatin that is inside the nucleus. And you have in regions of open chromatin, you will allow transcription to occur. And then you are going to have histone modifications that will wrap up the DNA and prevent. Then you will have these loops forming between different regions of the brain that allow enhance a region in the genome to go and regulate somebody that is really far away from them. And so those are epigenomic chromatin folding, where you have an enhancer that is megabases away from a certain gene, and by looping the DNA, you may get nearby the transcription starts at the promoter region of that gene, and that's the way it controls, even though you have a huge loop. So that is what is called high C.
Are these enhancers non-coding material in the gene?
Yes.
Look over.
Yes, enhancer regions are usually places in the DNA, short sequences in the DNA that have a regulatory function. They don't code. It's just like the promoter region, which allows the RNA polymerase to bind to it and start the transcriptional process. The enhancer or repressors are regions of the DNA that will enhance or repress that transcriptional machinery. But they are not, they won't code for a protein.
Do we know what causes the looping in this way so that the enhancers are now non-coding?
Yes, is depending on CTCF motifs and cohesing, it's looping regions of the DNA with a certain sequence. And then another protein that is like a ring that goes around, and when it finds that, it joins the DNA, and so it leaves a loop. It's but I'm not getting to that. Yeah, but it is known.
I'm glad it's also simple.
Yeah, it is known. And so what are the different layers that we can study? And so I put there the most traditional techniques that are used to study. So if you want to look at histone modifications, which are the ones that form the nucleosomes here, so all those histone proteins in the DNA where the DNA wraps around, those ones can be studied. They have several different modifications that are very important for the machinery that recognizes whether a gene is going to be transcribed or not. And so you can use ChIP-seq. ChIP-seq, all it means is chromatin immunoprecipitation. So you have an antibody, again, one of this histone, modified histone, you put it in your DNA, that antibody will bind to the protein that is bound to a piece of DNA. And then what you do is you clip and bring down that protein, and you can sequence that piece of DNA where it was bound. That's what ChIP-seq. The seq part is when you sequence that piece of DNA. Then chromatin accessibility is when you open chromatin, you are going to be able to access it with specific enzymes. If it is wrapped and compact, the DNA one will not be able to access and clip it. And so you can do a DNA treatment, and you are going to get these pieces of DNA, which only if they were part of open chromatin are going to be cut, and you can sequence them. And that way you can form a map of where those active regions of the DNA are. Then with RNA, it's again the same. You can sequence the RNA and know which RNAs are being produced in the cell. So coding RNAs, messenger RNAs, non-coding RNAs, and even what are called eRNAs, which is from those enhancer regions, little snippets that code for nothing, but just because it's open chromatin, it gets read. And the positioning of the nucleosomes, you can also study. You can study distal reaction using the different high C or 3C, which what it does is isolate these regions that are very far away, and you can sequence where. Because imagine if, say this is the DNA, sorry for the zoom, you may not see me, but here you see me. And so you have an enhancer region here, and you have a gene here, and this is 100 megabases, and you're looping it this way to be able to control this part. You have a way of isolating, cutting there, and then isolating this little part. And that way, when you go back to the sequence, you know this one belongs here, and this other piece belongs there, but now they come together, so there was a loop there. And finally, DNA methylation, which is what I'm going to talk to you more about, and then we are going to go a little faster. So DNA methylation refers to the addition of a methyl group on cytosines in the DNA. The place where these are put, and in general, is a repressive mark. So when a cytosine gets one of these flags of methyl group, it cannot open the chromatin if you want. And so in most species, the CG methylation is very high, and it's established in when the cells are dividing, and it's about 80%. So it's a cytosine followed by a guanosine, and that cytosine gets methylated. But it can also occur in what is called non-CG methylation, or MCH. We call it H because it's easy, but it's anything but G following the C. And this is more selective, and it occurs in certain tissues, like stem cells, the heart, I think some muscle, very low, and very high in the brain. And so using this, going back to the brain and its epigenome, we found a decade ago that both in mice and in humans, during development, we were analyzing from both species the regions of the frontal cortex. And we found that while the CG levels are maintained at around 80%, they vary, but it's not enormously. We discovered that this second form of methylation increases enormously, coinciding with the development of the brain. And it occurs mostly in the perinatal period up to adolescence, when it becomes a stable mark in the DNA. And we demonstrated that is depending on one of the enzymes that puts, which is called DNA methyltransferase, that puts this cytosines, this methyl group. And also, this is a plot, it's a k-means plot organizing the whole genome in what is called differentially methylated regions. So you don't need to understand much on this, but just to look at what is happening as the brain develops through life. And so here we are up, this is mouse, and we are looking up to 6 weeks of age, which is more or less when it becomes a adult brain, or they are able to mate. Let's put it that way. And so you can clearly see there that there are regions in the genome which lose their methylation. So the DMR here is blue, which means low methylation, while early on in development, methylation was high. And at the same time, there are others where methylation was low. And as you develop the methylation, so this implicates that this DNA methylation in the brain, and mostly is in neurons, which is what these cells that we isolated there are. We are able to separate neurons from non-neurons and analyze the methylome. And we saw that basically the ones that were really changing were the neurons as the brain develops. And so in a sense, CG methylation is not that stable. It turns out it's quite dynamic as you are developing and you get to adulthood. And so this was the first indication that DNA methylation patterns are cell type specific. They are not the same in all cells. And so that gives us the idea, okay, this might be an epigenomic control of cell identity. And so
I'm sorry, the last column there, the neuron N negative, what does that refer to? The
this
last column,
NUN,
the N negative,
non-NUN positive, so non-neurons.
or non-neurons.
Yeah. Neurons are here. It is the way we isolate the nuclei. It expresses NUN, which is a protein only expressed in neurons, or it does not express. So what we do to do these studies is we take the chunk of brain, homogenize, obtain the nuclei of the cells, because in the brain, in other cells, in other organs, you can isolate the cells themselves. But in the brain, the cytoplasm of the cell is the processes. And so when you try to isolate, you sever those processes and you change the RNA content. And so the best thing is to isolate nuclei. And neuronal nuclei express this protein. Non-neuronal nuclei do not express. And so that way we can separate and analyze the two groups separate. And that's the way that we discover methylation patterns seem to be cell type specific. And then my colleagues went beyond that, and I'm gonna skip that, and analyze the methylation patterns. This is just a way of showing. Each of this is a C. When it's blue, it's a CH, so it's non-CG. When it's green, it's CG. And the little bar means it's methylated or not. And the underlying gray line is the genome. And so you find these little bars across, for example, we are in the genome in the region that codes for the LHX6 gene. And the way it's shown here, the coding is in the inverse trend. So it goes from here that way. The green blocks are exons and the gray bars are introns. And that's the normal way that a gene is shown in any genome. Okay, and so when you look and you separate, we have ways of separating cell types in the mouse brain, which we cannot do in the human brain, but in the mouse we can do. And when you go look, the top part is a RNA expression. And so you can follow there the epigenomic control. So in this side, this SLC6A1 is a receptor or a transporter expressed only in inhibitory neurons. And then we go look, where is it expressed? Is it true that it's only inhibitory neurons? It's so small here that I'm going to try to do it. So here you have the whole cortex. You have a very small expression. This that says X are excitatory neurons, so non-inhibitory neurons. And these are two types of inhibitory neurons. So you can see very clearly in the excitatory neurons, that gene is not expressed. However, in the inhibitory neurons, it's highly expressed. This is a transporter that picks up GABA from the interstitial space. And when you look at the DNA methylation, you see it's highly methylated in the excitatory neurons, but very lowly methylated in the inhibitory neurons. And when you look at MCH, was a surprising part. In the NCH content covers the whole gene. So what we realize is that when you don't find methylation in a certain gene, that gene can be expressed in that cell type. If it's fully methylated, that gene will never be expressed. And so this further confirmed that these patterns were cell type specific. By this time, the Brain Initiative appeared. And as you all know, is the brain research through advancing innovative neurotechnologies or brain. And the idea was to try to tackle the brain. And the first iteration of it was a small grant or call for grant applications that were related to developing the technologies to be able to study the brain. So we were in the very beginnings, and that the plan, the Brain 2025, which is not that other thing 2025 that we hear a lot now, was the first 2 points were to identify and provide experimental access to different brain regions, but identify the cell types that conform a brain. And so 10 groups were awarded, and we were lucky to get one of these grants from the BICCC to develop the technology to be able to do single cell genomics, to try to understand how those methylation patterns in each cell type, and that way do a taxonomy of all the cells that are present in the brain. And so why is it important? Because when you mush a tissue and you produce the whole thing together, the signals that you would have from every cell type come out in one single. You're not able to differentiate. So as I was showing you, when we mushed the brain, all the nuclei together, we found all these changes. When we separated between neuron and non-neuron, we started seeing, oh, they are not the same. And as you go down, you start finding different patterns for each of them. And so single cell technologies was something that was needed to be able to analyze and do a full taxonomy of the different system. And so with that grant, we managed to develop 2 technologies, which was a single cell methylation and single cell open chromatin. That was in collaboration with Joe Ecker and Bing Ren at Joe Ecker is at Salk and Bing Ren is at UCSD. And so once we developed this technology, the next phase was, okay, let's go after the mouse brain. All this, the idea behind in the Brain Initiative was to develop technologies that would allow us to now go to the human brain. So the first thing we did was map the mouse brain fully at single cell resolution. And I was involved in this is what it took. 250 brains sliced at 600 microns, dissected every region of the brain, isolate the nuclei, and do as I always say, it doesn't come in color. It comes this way. And so, sorry. It was me, the one doing all the dissections, because my people would look at me and say, no, I'm not doing that. We went there. And so then we isolated again. By sorting, we isolate the two populations of neurons and analyze them separately, single cell. And used, we analyze it based on DNA methylation or open chromatin. So open chromatin first, and these are what open chromatin allows you to look at all what is open and being active in the genome. And it will give you peaks of sequence that allow you to position where in the genome is being active. And this technique was developed by Behrens and Sebastian Priso, who's there, and many others. And all this, you can imagine, is like having the information that you see in the sky. And then you have to do something with it. And so this is a group, it's a very large group of bioinformatician data analysis people that through Lubine clustering are able to separate the different types of cells. And so what you are seeing here is just for neurons, and inhibitory neurons are here, the group. So the first class, inhibitory or excitatory, and then beyond that, you can start separating the different cell types that you have in that mush of a brain. And so using this technology, you get to the what I call the Jackson Pollocks, which is a cluster analysis that allows you to separate what you have in that mixture. So it's looking at the sky, bringing all the stars and determining they are all stars, but what type? And so then you use this type of analysis to separate that uses near neighboring and a very complicated analysis and marker genes to identify each of. And you can do this by analyzing open chromatin, just open chromatin, to know where in each cell type is it open, or you can use a DNA methylation. And using this, you can see this is an analysis of all the cis regulatory elements. So call it enhancers, promoters, etc., that exist in the genome. And you can do this clustering and separate by each cell type. And as you see, the open chromatin gives you an identity of each of the nuclei you are analyzing. So it means that the cells have a molecular identity in their genome that says, I am a layer 6 neuron, or I am a inhibitory neuron of the parvalbumin type, or etc. And then in subgroups, for example, this group, this somatostatin cell type, you go look at the cluster and you realize you can even subcluster it. And then you find all these different types of neurons. Some of them might be state specific, not true different cells, but they are separating themselves. And then when you go and look at the motif enrichment, which means you get all those snippets of DNA and you look at the sequence of it, and you find out which transcription factor would bind in that sequence. You can do a whole analysis that way. And there, you find the motif enrichment, and is one of them was KLF5, but this is just an example of how well in that little nucleus things are being coded. And then you can interpret non-coding risk variants from human orthologs by moving back.
Obviously, this analysis is super complex. So I'm wondering if you folks are utilizing AI in interpretation.
Yes, we do. We do, but not the ChatGPT. We do deep learning models, but they are usually designed to do this analysis. We have used deep learning modeling systems way before ChatGPT came about. ChatGPT is kind of the culmination and the ability to make these deep learning models available to everybody. But it has been used in science for particular. So each data set, for example, you can apply different models to analyze it. And in these ones, many are of that kind. So
I'm curious, the results you're getting from your AI methodology, is that enlightening your research team? Is it giving you ideas you hadn't even thought of?
yes, it does.
Interesting.
It does.
Interesting.
It does. Because it's based on the data. The thing is that the deep model is deep learning models are able to find associations that for us would take our lifetime to
Really?
find. And so, yeah, because they can handle much more information at the same time than what we can. So in that sense, I think it's fantastic. We use it a lot because you can have a machine, a computer handling information that otherwise would be very difficult. Think of astronomy. They have been doing it forever. There's nobody sitting down there and saying, this is A, this is B, this is C for the whole galaxy. But that type of modeling has been used. The thing is, it had not jumped into the normal public use.
Does it require like fact checking now or fact checking or?
Yes, because you are giving the
Gotta make
data.
sure it's.
Yeah,
Right.
you are
Or
giving.
hallucination.
Yeah.
Yeah, you cannot hallucinate, but you are giving it the data. And you are asking the system to find relationships in the data. And then you have, if it does a prediction, you have to figure out a way that is different from that one to confirm that the result is correct. So you always have to, you cannot believe.
Yeah.
In a sense.
So-
You get a result and you say, we usually say, how can I prove myself wrong?
It has to be falsifiable.
It has to be. And so if these conditions don't occur, then I should get ABC. But if I don't get ABC, you start saying, okay, something is not quite right. And then you try different modelings. And in fact, in that first 2013 paper, when we discovered the changes in methylation during brain development, producing the data took us about a year. Analyzing the data took about 5 years because we just ran into walls where we realized, no, this is not right because it's telling me the brain is a finger. So it's not. Let's start all over. And so is the data analysis in these cases is nowadays, from 10 years ago, we are much better at it, but we still keep on hitting the wall.
What you just said made me think there's a clear distinction between observation and function.
Mm-hmm.
We can observe how A, B, and C are existing. They are there. But how are they interacting?
How they are interacting, how they are expressing themselves, etc., is different. And the results can be
surprising.
Surprising, and many. And so we did the same type of analysis, but using the one before was open chromatin, and this was DNA methylation. And again, the system consists of separating neurons from non-neurons, and then do a modification to which is a chemical reaction with bisulfate. So all cytosines that are not methylated will be converted to a uracil, and then to a T when they are red, amplified. However, the cytosines that have a methyl group covalently bound to it will remain as a cytosine. And so then you sequence, and your genome will be a lot of T's, because the cytosines that were not methylated were converted. But you can map where the methylated cytosines are. And so that is the bisulfite conversion there. And then we put it in a robot, and we put in each of the 384 well, in each well of a 384 well plate, we put a single nucleus. And so we ran an enormous amount of sequencing on single nucleus, and then we go back and do the analysis that I showed before. And here is just an example of how you can separate on different levels. So in this case, you can separate by cell class, where you have the excitatory neurons here, the inhibitory neurons are in orange. I think these are the glial cells, and this is the hippocampal formation. And then you can pick up the inhibitory cells and do a second level clustering, and you start finding all the different inhibitory neurons that exist in the brain. And there you see that the striatum, the MSNs, all the different type of GABAergic cells start separating themselves in these maps. And then even in one cluster, for example, the striatum MSNs, medium spiny neurons are called, that express D1 dopamine receptor, can be subclustered in at least 4 types of those neurons. So as you can see, as you go down and you get a bigger magnifying glass, things look quite different. It's the same when you look at the sky with your naked eyes and you see a lot of stars, and they are your well-known friends, and then you decide to get the binoculars and you say, oh, there are a lot more. And then you go in a telescope and say, ooh, I cannot recognize anything there.
So I would think you wouldn't have been so surprised to have found the reward system dopamine factor within the striatum, because it's known those factors are functioning within the striatum.
Yes,
So I
but
think that would have
there are
been
several types that within the D1 MSN, there are several distinct types.
Okay, all right.
That we didn't know.
Okay.
And so you start pulling apart now the different cell types that exist in that brain region. Not just, D1 and D2.
Right.
We knew that there were
Right.
D1 expressing D2, dopamine receptor 1, dopamine receptor
Right,
2, and that we knew.
Right.
But within those groups, there are many more. And that's the important thing, because now you can go back to those cells and start asking, are they all involved in doing the same? Or this subgroup is connecting here and this subgroup is connecting there? Why is that important? Because, for example, in Parkinson's disease, where you have a deep brain stimulation, you go down there and you stimulate everybody inside in that niche. Not just the
Not just the
target cell.
dopamine. You want to
No.
regulate dopamine, not the
You
entire
want to
process.
be able to target the cell that is involved in the function you want to modulate,
Yeah.
not the whole region.
Specialization, yes.
Mhm.
Differentiation.
What, Zoom figured out?
We lost the, yeah,
Sorry, did I do?
we might have hit something, but that's fine. Let's see if we can get it back.
I have a question.
The last
You
one-
Pay attention there.
Yeah, now it's okay.
Ask this question, I will. It's kind of, maybe off topic, it might, but it doesn't get, just might be a meta topic. To it. And, it has to do with all this differentiation and the possibilities that creates in functioning. And it really comes out of a meeting we had yesterday, where, seeing about how the interaction of waves and the cells, becoming excited and creating.
Yeah.
Oh, I'm sorry, we're back.
Yeah, but
Right.
we got people
I
talking.
had a question though about this. Does that differentiation of the D1, does that mean that there are 4 subtypes of D1 receptors?
At least.
Wow.
You see, you can see it here. These are markers. Different. They are expressing different proteins,
Yeah.
each of the clusters. You can differentiate and separate them. So that is what this is bringing about, that you're starting to get a really good idea of, in that brain region that you go and whack with a deep brain stimulation, which cells are there and what is their behavior when you go and excite them all at the same time. Because if you are thinking about cortex and you stimulate, for example, the prefrontal cortex, you have all those different layers of excitatory neurons, and then you have inhibitory neurons that are going to react, and you have astrocytes, and you have the vasculature. All that can, the response might be different. And so this is just the similar, but for the excitatory neurons. And there you have this big blob here. They are in the first layer of separation. They all have a commonality, and so they fall into that excitatory neuron class. But then you can even separate them, not just by the layers. So you have here the layer 6, layer 5, layer 4, but also on the different brain regions. I didn't put, yeah, you have it here. These are all, this is olfactory region, piriform cortex, orbitofrontal, prefrontal, the insula, motor cortex, somatosensory cortex. And you can see they separate themselves. So even the brain region, it has an internal molecular identifier that allows you to separate them. And so this led us to have to further on the development of technology to be able to, instead of doing a separate open chromatin one side, DNA methylation on the other, and then try to put the two together, it proved to be very difficult to integrate those data sets. So we had to develop ways of determining both in the same nucleus. And so that's what we did. And so we invented this SNM3C, which measures DNA methylation and chromatin structure in the same nuclei, or a pair tag that is able to look at RNA expression and histone modifications in the same nuclei, or there are many more of them. And using these different technologies, I don't want you to understand anything of that, but this is just the way that you can parcellate the whole brain and then go look at the this is the whole brain, the whole mouse brain now. And you have the clustering analysis. Here are all the different classes and subclasses of almost 5,000 cell types in the brain. And then you can look at chromatin structure the same way, and that it is also cell type specific. So those enhancers that I was showing you with this are also defining the cell types. And now we can have all these different data sets put together to integrate and make definitions of each cell type with very, very deep understanding of how the cell's identity is. And then, of course, you need to go back to the brain because the Jackson Pollocks were very, very nice. And so what we use is a technology that is called MERFISH, which what it does, I always call it is I don't know if you know what in situ hybridization is. It was used to be used for detecting RNA species within a cell. And so this is like in situ hybridization. It used to be 1 by 1. This is on steroids. So you can do 1,000 genes at the same time. And that way we can pick up from our clustering analysis, pick up the genes that we consider are the ones defining the cell types, and then go back to the brain and look where are they expressed. Are the predictions done with the models corresponding to what happens in the brain, where those cells are. And so this was all published in 2023. And so given that, we were given funding, and that was the big grant that we got to the human brain. And, oh, sorry, that is his son, this one. He worked with me, Nick. And so we started now with the human brain, and what we are doing is a similar study that we did with the mouse, but 125 regions of the human brain, where we are determining the cell taxonomies of all those regions. The idea is, many people ask, well, you have already RNA to identify cells. Why do you need these fancy technologies that are really expensive? Because RNA doesn't give you the regulatory regions. And it is the regulatory regions that will allow you to modify the function of the cell. As we have recently seen the correction with CRISPR of that mutation in the baby that would lead to his death. And now the baby is the necessity of having a very clear understanding of the regulation of the genome in a particular cell type. And these technologies allow you to map that. RNA will allow you to map only those genes that are being expressed at that point in the cell. These other ones are telling you how the regulation of that transcription is happening. And so we were funded to map the human brain at that level. And as a pilot, we did DNA methylation and chromatin analysis. I'm not going to talk about that because then it's too much, and I want to talk about aging. And we did a comparative single cell atlas of chromatin accessibility and histone modifications that allow you to separate the different cell types in the human brain. And so, yeah, what do? We buy a lot of computers. Right now, we are in the cloud, but NIH has a system where we put all the data because it's really, really a lot. And the analysis takes an enormous amount of computer power. So we are basically doing it in the cloud because this became too big. And so one of the interesting things that we analyzed in the human brain was, can we go now look to see if we can assign the cell types to those single nucleotide polymorphisms that come from the GWAS study associated with specific diseases. And so we said, well, we have the single cell data. We know which cells have this or that enhancers or cis regulatory elements. That's what CRE means. And can we give a hint of those single nucleotide polymorphisms that, give an enhanced risk for depression that have come out from those GWAS studies? Can we assign the cell type where that would be important? Because if that single nucleotide polymorphism falls in a region of the genome which that cell has totally silenced, cannot care less if it is the correct base or a wrong one. But if it falls in a region that is active chromatin, it might affect the expression in those cells. And so I'm not gonna. And so this is what we found using open chromatin. And as you see, there are groups of cells that are related to where you can pinpoint where the changes might be important. And so for schizophrenia, we find that in cortex, excitatory neurons and inhibitory neurons as a whole, there are single nucleotide polymorphisms all over the place that tell you those two cell types might be important. But look at Alzheimer's. It's microglia. So the GWAS study, the single nucleotide polymorphisms, target directly a change in the microglia cell type, not in all the rest of the cells. And so this is the power of having the information on those enhanced regions that are far away from where the actual gene is. And we find similar results analyzing the data from the methylation point of view. So methylation will shut down genes. So if we go and look at the methylation state of enhancer regions, we surely are going to find that if it's methylated, it's silent. And so if the single nucleotide polymorphism change, for example, a base to a C followed by a G and it gets methylated, that region is going to get shut down. And it's not going to do the function that it would be doing in the normal. And the same happened with Alzheimer's. Microglia are the ones showing. These are, you would say, sporadic mutations in certain people, but the GWAS studies are done with 100,000 or more people to get the power to find. And so that all that technology that we developed allow us now to look at the changes in a very specific, these are all the people that were involved in this study, but what we looked at was sex and age differences in one specific region of the brain, which is area A46 or the prefrontal cortex. And so what we looked in this study was whether there is a variability within individual in the region that we were looking, and also the inter-individual variability between subjects. And so for that, we used the technology that we developed also that is able to analyze DNA methylation and RNA in the same nucleus. And that is the publication. And so what we did was analyze males and females in between 20 to 25 years of age or 70 to 75 years of age and compare. All coming the same region of the same hemisphere. And we do nucleus sorting and personal genome because you need that for being able to map back. And analyze RNA and DNA methylation. And so from that study, the cell types, this is like a QC of the thing. The cell types we find in every individual are equivalent. So we are dealing with the same cell types. And then we look at how whether the DNA methylation followed the patterns that we had discovered before, which is when it's hypermethylated, the gene is not expressed. If it's hypomethylated, the gene is expressed. And that was the case. And then one of the things that we found was that there was not a true intra-individual variability, so the whole region did not show. What we did was for the area 46, which is quite big, we took samples from the two edges of it and compared to see if there was variability. And we didn't find this variability, but we did find across all individuals inter-individual variability, mostly on the outer layers, the top layers of the cortex. So from layer 2 to 4. There you find high variability. If you think about developmental wise, those are the last ones to mature. And it's possible that the variability we are seeing is related to experience-dependent plasticity that is less pronounced in the deep layers of cortex, but yet to be.
But that should be different in the older versus the younger population.
You would expect. We did find a lot of changes in the older population. But those initial ones that may be defining this are that when the methylation patterns are really dynamic, which is between birth and adolescence, may be the ones giving that pattern.
These are all adults.
Those are adults, 25 and up. So up to 75.
Are these like people that died?
Yeah, postmortem.
Okay.
Yeah.
Yes. Those are why no, we don't get it. What? That's not true. You can get tissue, healthy tissue from tumor resections, from when they take out for ablation for epilepsy. You can get healthy tissue where you can do studies, but it's way more difficult. This is all postmortem brains that have been donated to biobanks. And that's where we get the brains. Yeah. I'm not not looking at people, although we are, because we are having a lot of trouble finding healthy people. So what we found overall was that there is an increase in DNA methylation as you age. And about in most of the neurons, there is what you would say a shutdown of the system. And what we found is that the inhibitory cells reduce in numbers. So, and that is, it was something known, the somatostatin and the VIP cells, the number of cells present in the all brains is lower. This coincides with studies in Alzheimer's disease, where they also find this, and we were discussing with the author of this paper and saying it could well be Alzheimer's disease. What it does is push the aging process to younger ages with respect to this loss of inhibitory cells. And although the inhibitory cells themselves disappear in number, you still have, but they are less numbers. The ones that had a more pronounced effect from RNA and methylation were the excitatory neurons, in fact. And here are the changes in transcription and expression, and most of the genes are related to synaptic structure and function, which is something that has been proposed before, but this is like checking out which genes are going down. And also in studies that we have done in mouse, we have found that there is a lot of changes in transposal, the line elements, and that it appears highly in. And then there is a very clear downregulation of an activity-dependent gene, which is the EGR1 from excitatory neurons that is related to the ability of fighting and maintaining that structure in or function in the brain. And the excitatory neurons show downregulation of those genes. So in a sense, the neurons are there, but they are linked. However, not all the genes are downregulated. There is the glucocorticoid receptor called chaperone, the FKBP5, which is strongly upregulated in age neurons, and this gene is involved in stress, in inflammation, etc. So in a sense, it's not different from what other type of technologies have suggested for the age brain. And
Do you folks have guesses for the cause of this neuron reduction?
no, I would not go there because there are many reasons why this could be happening. But one of them, who's first, the chicken or the egg? Is it the functioning of the neurons that lead to changes in, for example, free radicals that tend to affect the DNA, and that is changing? We are deeply studying more functionality of this is description of what is happening to the neurons. And what is driving it, I would not.
Is one, and I've been thinking this lately, is one guess a lack of oxygen and blood flow to the brain given aging? Is that a factor?
That could be a factor, and in fact, it would be nice to be able to now compare that between different brain regions, because one of the regions of the brain that is most affected by the drop in oxygen is the prefrontal cortex. So other regions are not as affected as the frontal part of the brain that the That's one of the frontal dementia is related to strongly from that hypo oxygen in the brain, mainly when people sleep, and that's why they have to use the oxygen. And so not all brain regions suffer the same way with less oxygen. So the interesting part would be now to go and look in the different brain regions, how in an aged brain, how they change and whether you could correlate. But then you would have to do experiments to show, to prove that that is the case, not just a speculation. So we know there is a lot of things that we know, and it could be related to that, but I wouldn't say go put your oxygen mask. But if you have emphysema or things like that, do put it because it does affect. Normally, as you sleep, the oxygen, under normal conditions, the oxygen pressure in the brain is lower than outside. And when you sleep, it goes even lower. And so that is the reason why if you have problems with oxygen levels, it's a good idea to have the mask as you sleep to prevent that really drop that will affect the functioning of regions such as the frontal cortex. And one of the things that we found was very interesting is that the subtelomeric genes, which are those ones that are at the edge of the chromosomes, tend to be upregulated. And from studies in the mouse, we found also that in these regions is where the retrotransposons get unmethylated. And so this is, you can see here in the telomeric regions, the genes that are nearby the end of the chromosomes tend to be upregulated. And this is related to something that is known about cells when they are getting old. They tend to shorten their telomeres. And so that is giving alteration in the genes that are expressed nearby there. And so there you could have a cause. Genes that shouldn't be expressed in those cell types suddenly, because of the shortening of the telomeres, they start being expressed and affecting other genes in the genome. So in summary, for the aging study, aging has the distinct effect on neuronal cell types in the human frontal cortex. The GABAergic subtypes of somatostatin and VIP are dropped in numbers. The upper layer intratelencephalic IT glutamatergic neurons that I was showing you have hundreds of differentially expressed genes and are the most variable among individuals. The deep layers are way more compact. And there is reduced expression and hypermethylation of synaptic genes, which would slow down the activity of neurons. And there are things that I didn't talk about, sex difference in the chromosome, but it's been a long time. So different things are. So these are all the collaborators across, and of course this is a work that involves an enormous amount of people and very strong collaborations between groups that are the skills are very different, and so genomics, neuroscience, and DNA methylation, So it's a very large group of people, and also the whole group of in the Brain Initiative, in the consortium.
Well, I have a question, and that was the association between Alzheimer's and microglia. If I remember, are microglia immunologically
Yeah,
active cells?
microglia is called the immune system of the brain.
Well, I have a question, and that was the association between Alzheimer's and microglia. If I remember, are microglia immunologically
Yeah,
active cells?
microglia is called the immune system of the brain.
Yeah,
Yeah, and so those microglia are the ones that respond and produce cytokines, and are the ones that clean up, if you want, the brain. They really behave like macrophages and that. So they are the ones that go and prune synapses, etc. But it's in a sense, when you get activation of microglia, you usually have, if it's a strong activation, you usually have an inflammatory process happen.
and so when you were looking at the ranges between old and young, did you see differences in the microglia that way?
We did not look at that population, unfortunately. But not what that data was saying is that there is a single polymorphism, which means one base in the DNA is changed. And that change correlates very strongly with Alzheimer's disease. So you are more prone to find that little mutation in people with Alzheimer's disease. And that we just brought that information into the single cell data that we produce to particularly say that single nucleotide polymorphism will affect enhancer regions active in microglia. And so although microglia is known to be related to inflammatory process and being active in Alzheimer's, what we bring in is that sporadic mutation that has been associated with increased incidence of Alzheimer's is going to be active in microglia. The other cells are, that region of the DNA is shut down. So it doesn't matter if they have it. All the cells will have it, but it only functions in those ones.
Got a couple of questions on methylation. So when you get a hypermethylated cell, is that because the methyltransferases are upregulated, or just the targets are more accessible?
Both. When you are during development, is because the methyltransferases are activated.
Okay.
In the normal brain, there are many things that regulate the activity.
Yeah. The other part of that would be in a developing situation, you said that I guess methylation is one of those features that is mitotically heritable.
Yes.
So does that mean all of a lineage of a developing cell would have the same methylation pattern, or would it change if you develop into a different type of?
They change, but you can go back to find the original one because there is vestigial DNA methylation that comes from and that's the way that you can go from, for example, the DNA methylation patterns that we use to identify cells in an adult brain. When you go to a newborn brain, they're not there.
Right. So, yeah.
So you have to go back and look which are the mitotically defining ones to be able to trace them back.
I'm curious, number one, how hopeful, and number two, what your projected time frame is. for the Brain Initiative's research to yield effective treatments for, if not all, most of the significant brain disorders, including Alzheimer's.
5 years.
No, well, that's what it's gonna, well, if it doesn't stop this year. You are creating a knowledge, an atlas of the brain. Only then, because the Brain Initiative, the section I am working in is the cell senses. There is a whole branch studying the connectomics. There is a whole branch looking for ways of delivering into the brain, ways of delivering drugs, delivering virus to modify. And so it is the joint of all this is going to be the one that is going to give leads to go analyze what you are saying. So we are in the very beginning. So it's just like the human genome is going to be the cure for all, and we are still mapping the human genome. And it hasn't been the cure for all, but it has taught us a lot about what to expect. And so on this one is we are creating an extremely detailed map of the cells in the brain and how they connect to each other and what regulates them. And now you can go with that knowledge and start looking for specific diseases of the brain and how you can use all this data to be able, for example, to deliver in glioblastoma, to be able to deliver a type of a drug or that will affect exclusively the glioblastoma cells and not any of the healthy cells that are around. Or if you want to modify a cell type that is inactive. For example, in Parkinson, you have cells in the substantia nigra, which are the dopaminergic cells that die. Those cells are the ones that signal to the striatum and from there to the motor cortex. Okay, you start losing the connection, the stimulation, if you want, or the regulation from the VTA. But if you have a good knowledge of which are the cells that came from the VTA to the striatum and are controlling the motor cortex, you can stimulate only those ones and get, because the rest of the cells are fine. So you can go much more detailed on which is the pathway you want to activate without touching everything else. Or in Alzheimer's, we need to first understand what triggers it. What is the cause? Is it an activation, weird activation of microglia at the wrong time of life that starts changing the way amyloid beta and tau protein get processed and you get the tangles? Are the tangles and amyloid beta deposition the consequence of something else going wrong? We are still trying to figure out that. But if you start learning how, in which parts of the brain and which are the cells most affected, you can start having, even though you have the plaque there, you can stimulate the cells such that they keep functioning. Because remember, what you have in the cells you have in your brain are the cells that you're gonna live with. All you do since you are born is kill them. Fortunately, we have a lot, but they don't reproduce. And so the neurons you have by age 90 are the ones that you saved, in a sense, but you're not able to create more. So what you want is not to rescue a cell that is half dying, but try to stimulate the network. You know, when the whole comes in, it's like, how do you jump it and substitute the function of that cell that is dying? Because recovering a neuron that is about to die Might not be a good idea because many things have changed in that new.
Do you have enough technological equipment at present to fulfill your mission? Or over time, are you hoping for new technological equipment to be developed?
No, we need more technology. Yeah, we can answer certain questions now. But as during these 10 years, as we were going through, it's like, we're gonna go, we need to develop new. And that is the way, and as soon as some other technology comes in, you try to adapt it to see if you can answer more questions. But yeah, technology development is always necessary.
We have a question from the
Yeah.
Zoom audience, and it says, I notice on the list of interpreting non-coding risk variants, there is a category of insomnia.
Yes,
How does this pertain to a human being?
no, it's all humans.
Yes.
That data is
So
from human.
do some of us have a tendency toward insomnia?
Yes, there is. There
Okay.
is. And so there are those variants have been associated in the overall population with insomniacs. There are some that is genetic, they know very clearly the mutations, but there are others that are non-coding variants or single nucleotide polymorphins that give the tendency in that people to be more insomniacs or have more trouble.
It's kind of related, I think it's directly, same kind of question. Is your analysis now able to more clearly define when you have multiple genes involved in a phenotype like insomnia? In other words, I think they're called quantitative trait loci.
Yeah, yeah, it would. You get the information from the QTLs, but then what our information gives you is in which cells that is important.
Which cells, not so much which genes.
Not much which genes, but if those QTLs are in a specific cell type, that cell is going to be the one manifesting the brunt of the change. And for us, that is important because it's a way, okay, I need to be able to go and tickle that cell and not all the rest. And so the dream is to be able to, for example, do a CRISPR modification of a cell type that is producing a certain effect. So you can change the DNA and now that cell should be able to locally and only those cells. And for that you need to, and there is a whole part of the Brain Initiative is all this information we have produced is now, okay, which are the cell type specific enhancers that we can use to deliver, for example, a virus with a CRISPR that will affect exclusively those cells, or for the people using electrophysiology and optogenetics, for example, how can we make this very specific cellular technologies for analyzing activating, inhibiting certain neurons without affecting the ones nearby or the ones connecting to them.
Just in terms of, this might be the last question. So in terms of, from an evolutionary perspective, I mean, the machinery of this epigenomic accessibility is amazing. Was that machinery there very early on in invertebrates, for example? Do they methylate their?
Not all, not all. For example, which one is it? C. elegans doesn't use methylation as a regulatory. They use no-coding RNAs. But the majority of mammals, and if not all, use this mechanism as the way to regulate the genome. But not all. I think ants don't, bees do. And so it's kind of a
But
special.
so is there a sense that the complexity has increased up to the human level, or do we know? Like between the mouse and the human, are there things that the mouse is unable to do that humans are able to do?
No. It's very humbling, but even the cell types are very similar between the two species. There are a few cell types that are only present. No, not even. The von Economou neurons were thought to be only present in the human brain, and now they have found them in the mouse too. So it's very humbling, but yeah, we don't have whiskers, but some dogs. What we do have is way more of each type. And so that is the expansion. If you look at the mouse brain, that normally the dorsal hippocampus, which corresponds to the dorsal is the back in the human, but in the mouse is here. In the human is here,
Yeah.
because the cortex expanded enormously, and so you have way more food for thought. The area that you use for each of the functions is much bigger. And when you think about it, is that the cells, as you saw at the RNA or methylation level, is a cloud, what we call a cluster. They are not all in the same state. They have that ability to be in many different states. And so the larger, you have more capacity of having them in different states. And so if our brain was all composed of cells that do yes, no, it would be very boring. But it has that sort of large possibility of responding. Not all cells respond exactly the same. And so it opens a possibility for many responses of the brain region. But anyhow, speculation.
Thank you very much. Thank you.
But thanks to you.
Well, thank you.
Sorry, it took